All right, so now for something pretty different. Hopefully, mine's a little more wet lab based, but hopefully it'll at least whet the appetites for the modelers and theoreticians out there for thinking about what is actually going on in the brain that at least what we can see. So my project is looking at the physiological basis for emerging evidence of lateralization in the mouse auditory cortex. And so our lab has previously shown, this is work done from a former grad student in the lab, that the left auditory cortex is important for maternal behaviors. So when the left auditory cortex is inhibited, but not the right, mouse females that are trained to retrieve pups can no longer are less likely to perform that task when it's inhibited. More recently, another former grad student in the lab, Schiavo, recorded a library of pub calls and found that the prototypical pub call interval is around 150 to 200 milliseconds in between each pub call. And when that library is played to naive virgins in the left excitatory neurons in layer 2-3 of the left auditory cortex show a preferential response to that prototypical pup call interval. However, once these females learn to retrieve pups, their responses are broadened to a greater range of pup call intervals. So what we wanted to find out is if there was an innate mechanism within the layer 2-3 of the auditory cortex that causes this preferential response to pup calls at this 150 to 200 millisecond pup call interval. So what I did was I recorded from coronal slices of the left auditory cortex, patched onto the pyramidal neurons of layer 2-3, and used an electrical stimulus as a crude proxy for pup calls, and stimulated them at various intervals and measured their responses. So what we can see here is both the excitatory and inhibitory currents, inhibitory on the top and excitatory on the bottom. And what we found is that both excitatory and inhibitory currents depress at all the stimulus intervals that we measured. But interestingly, the inhibitory currents depress at significantly faster rates at the prototypical pup call frequency at 150 milliseconds versus the excitatory. And this is the case in the left auditory cortex of females and is not the case in the right auditory cortex. So what we wanted to do was find out if there was a specific interneuron that was driving this specific depression at this interval. So we measured both somatostatin and parvalbumin interneurons, driving them specifically with Cre-dependent channel rhodopsin. And what we found is some disparate results. The somatostatin neurons actually demonstrated a short-term plasticity that actually increased with shorter ISI intervals, whereas the parvalbumin demonstrated depression that was similar to what was seen with the electrically driven stimulus. What is interesting is that while this may not be completely novel, Barry Connor's labs has shown similar results in layer 4 of the cortex. But what is interesting is that neither of these neurons when combined can explain the specific depression at 150 milliseconds. What is also interesting is that there is no difference in the depression that is seen in with electrical between naive females and retrieving females. So that this short-term plasticity mechanism is not probably what's causing the broadening of the responses of females of female auditory cortex after learning. So we want to look at some other long-term possible mechanisms. So our lab has also shown previously that the auditory cortex can undergo plasticity both excitatory and inhibitory plasticity through the mechanism of spike timing dependent plasticity, as shown here on the left. And then Mike Hickley's lab has shown that in the layer 2-3 of the prefrontal cortex, somatostatin interneurons specifically undergo potentiation through NMDA activated currents, whereas parvalbumin and VIP interneurons do not. So we wanted to investigate this specifically within the auditory cortex. And what we found is that yes, in my hands, somatostatin interneurons actually do undergo spike time independent plasticity, as you can see here. But when we investigated the other interneuron types, namely the parvalbumin, VIP, and then NDNF interneurons, which are used to drive which are used to label a population of layer 1 interneurons that synapse onto layer 2-3 pyramidal neurons, that neither of these populations on average show synaptic plasticity. So altogether, what we found is that the somatostatin interneurons are privileged in the mouse auditory cortex, and then they both show short-term and long-term plasticity mechanisms. And the consequences of this belies an important role in auditory perception and learning. And what we want to do is to combine these results into a modeling study to figure out what are the actual consequences in learning for the auditory cortex. So I want to acknowledge several members of the lab, including my mentor, as well as the highlighted people that are in the lab member list. These are the co-authors of the paper that has just come out about a month ago. With that, hopefully, I think I probably talked really fast, but I'm glad to take any questions that you guys have.
Yeah, thank you, Soomin. It's just like, this is how we were planning to have more time for questions. You are the only one who did a short talk. If you have any questions for Soomin, please post them in the question and answer panel, and we will try to answer them. Did you expect, I mean, while we are waiting for other questions, did you expect these gender differences?
So I guess not necessarily the basic electrophysiological differences that we noticed. We had a hint of it possibly because of the fact that female mice showed this laterality of the responses, and then I guess we didn't expect male mice to do that as well. But we have some other evidence leading to some gender differences, but yeah, I guess not necessarily. We didn't expect to see it in the basic physiological differences.
And why do you think it? I mean, what is your speculations why this is occurring? I mean, I can't get any specific reason why there is a big. I mean, this is the interesting differences, right?
Yeah, no, I completely agree, and I mean, it's very rare to find gender differences, as well as very rare to find laterality in the cortex, especially not in humans. So I mean, maybe it's because the female brain is at least in mice are need to be attuned to these pup calls, and so therefore they have to be highly sensitive to the prototypical pup call for them to be able to learn the maternal behaviors, especially if they're not being trained by another dam or another experienced female.
Okay, that's interesting. I'm just wondering, is there I have no idea about that. I'm just like wondering the number of the SST or interneuron distributions. Is there any differences between females and males?
Between females and males, I don't know if that's done actually. So that would be an interesting question. I do know that there are age differences in the expression of the different neuron types, but I don't know anything about laterality or sex differences.
This is interesting. If you have any questions, please feel free to post them in the question and answer panel, as well as our panelists. If you have any questions for Soomin, please feel free to ask them.
Yeah, I had to cut out a lot of the methodology. If you have questions about the specific methodology, I have those as backup slides if you need them.
Please go for it.
All right, so basically, for the electrical, that's pretty obvious. But for the spike time independent plasticity, the methodology I used here was I used the pre, before, post only. I have not done the reverse, but that has been shown in our lab previously to work for specifically inhibitory inhibition. But I used a 5 millisecond interval between the presynaptic stimulation and the postsynaptic current injection, and I repeated that 100 times at a 2-second interval. Waited 5 minutes and then did my follow-up recordings. And what's interesting is that, so we've found that, at least in layer 5, that the inhibitory spike time independent plasticity is also NMDA dependent. So it is not presumed that the inhibitory interneurons is providing its own NMDA. So this is sort of like a heterosynaptic or maybe non-specific plasticity mechanism.
Cool. We have one more question for you from Bernard Slater, asking for how are you doing the between cortices comparison to show that the responses are different?
I see. So especially for the short-term plasticity mechanisms, I recorded this in left-right auditory cortex, naive and retrieving females as well as males, both left and right. And so it is only the left auditory cortex that has shown this specific differences. Males and right females don't show this laterality.
It's within the same animal, right?
Yes, correct. Within the same animal, yes. So I'll make slices, make sure to keep track of both whatever left and right auditory cortex, and then I would alternate recordings from either side to try to get within animal comparisons as well.
That sounds really good.
And thank you, Soomin. If I may ask a question before we end the session as well. It's really interesting. This kind of brings a deja vu for me. Of the research that was done on the fusiform face area, as it was called, or the fusiform gyrus, as it is called now, is that, a lot of us thought initially that the FFA is just accustomed to processing faces in humans. And then we, went on, researchers did more research. They found out that, okay, the FFA does other stuff in other mammals, and even other apes. It does other functions. And then they did higher, sample size research on humans, and they actually found out that, for example, furniture salespeople, they tend to process furniture in the fusiform area. And then, this is to bring context for everything. And then they, went on dog show hosts and dog show referees, and they actually found out that, they tend to process some things like canine teeth or some very specific things that they were highly specialized in that area. And, right now, there's more body of research that actually shows, okay, fusiform area is for specialty. And us humans, as social animals, we tend to focus a lot on face processing. So in smaller communities, you might not have as much focus on FFA for that. So would this difference, this lateralization, would that possibly be something like this, that it is actually the result of a flexible environment in the cortex that actually is giving rise to two different behaviors physiologically? Would that be possible in your opinion?
I mean, yeah, I would say that's possible. I mean, we do know that for mice, vision is probably the least of their sensitive senses, and therefore least used in the processing. So, but hearing is a lot more sensitive, and therefore would probably belie more specialization in their processing, and so therefore maybe you have more lateralization in that case. Yeah, I mean, it is definitely possible that especially for females that, like, in order for their species to survive, need to be able to care for their pups, and therefore they need to be able to respond specifically for these pup calls. And females that are not trained to raise pups need to be, vigilant for these prototypical pup calls.
I think at this point we should just summarize, and thanks to Soomin, Christopher, it's just very nice work and it's interesting findings. And I also would like to thank to all of our speakers, Melanie, Badr. It was a really beautiful results, and I can see your reason why you made the really long talks, because you wanted to share your interesting findings. And I also would like to thank to our audience for joining us in this session. I think we should also thank to Taha for giving us technical help and sharing our session during the session at the Twitter and making it much interactive. I hope you enjoy these sessions and hope to see you in the next Neuromatch Conference. And thank you very much, everybody.
Thank you, everyone. I'm going to end the talk in 5 seconds.
Thank you.
Thank
Bye.
you.
Bye-bye.